This lecture will cover pentose phosphate pathway, what it is, which reactions constitute pentose phosphate pathway, and how it is regulated. Pentose phosphate pathway provides electrons in the form of NADPH for biosynthesis of lipids. Just to remind you from chapter 1, electrons are needed for catabolism, that means synthesis of ATP, and they are provided in the form of NADH. For anabolism or biosynthesis, electrons are provided in form of NADPH. So that additional phosphate group on second carbon or ribose is a tag that indicates that that molecule is to be used for anabolism, not catabolism. Electrons are provided mainly for biosynthesis of lipids, but they are also used for biosynthesis of other molecules, such as 2-deoxyribose that is needed for biosynthesis of nucleotides. And pentose phosphate pathway also provides ribose for synthesis of nucleotides. The first three reactions of pentose phosphate pathway are oxidation reactions, and they involve production of NADPH. Remaining reactions are involved in production of various monosaccharides and are not oxidation reactions. The first reaction is oxidation of glucose 6-phosphate to 6-phosphoglucono-delta-lactone. That compound is unstable, while monosaccharides themselves are in general more stable in cyclic forms. Delta-lactones are not, and this particular delta-lactone undergoes spontaneous ring opening. In this case, it's catalyzed by 6-phosphogluconolactonase to make reaction faster, and resulting product is an open chain carboxylic acid. It's called 6-phosphogluconate. In the next step, 6-phosphogluconate is oxidized to 3-oxo-6-phosphogluconate. Oxidizing agent is NADP, and it's reduced to NADPH. That's how NADPH is generated in the course of pentose phosphate pathway. 3-oxo-6-phosphogluconate is a beta-keto acid. As such, it is unstable and it is decarboxylated. It is decarboxylated to ribulose 5-phosphate, so a keto pentose. When it comes to reactions that generate other monosaccharides, the most important reaction is isomerization of ribulose 5-phosphate into ribose 5-phosphate. Enzyme ribulose 5-phosphate isomerizes ribulose 5-phosphate into ribose 5-phosphate. Ribose 5-phosphate is important because it is starting material for synthesis of nucleotides. It is also starting material for synthesis of 2-deoxyribose that is needed for synthesis of DNA. 2-deoxyribose is produced by NADPH reduction of ribose. There are other reactions that are involved in the pentose phosphate pathway. One of them is conversion of ribulose 5-phosphate into xylulose 5-phosphate. That reaction is catalyzed by a phosphopentose epimerase. So carbon 3 is epimerized. Remaining reactions, here are some examples, involve enzymes transketolase and transaldolase. Those enzymes catalyze formation of other monosaccharides. Transketolase transfers 2 carbon units and transaldolase 3 carbon units. Donor molecule is always a ketose, and the acceptor molecule is always an aldose. Here are various examples, and monosaccharides with 3 to 7 carbon atoms are formed. In example 6, xylulose 5-phosphate, a ketose, is a source of 2 carbon units. That means that reaction is catalyzed by transketolase. Ribose 5-phosphate is the acceptor, and products are glyceraldehyde 3-phosphate or GAP and pseudoheptulose 7-phosphate. In the example 7, glyceraldehyde 3-phosphate, therefore an aldose, is the acceptor, and pseudoheptulose 7-phosphate, product of the previous reaction, ketose, is the donor. Since 3 carbon unit is donated, reaction is catalyzed by transaldolase. Products are fructose 6-phosphate and erythrose 4-phosphate. And finally, in the last example, example 8, xylulose 5-phosphate, ketose, transfers 2 carbon units, so reaction is catalyzed by transketolase to erythrose 4-phosphate. Products are glyceraldehyde 7-phosphate and fructose 6-phosphate. In the final part of this lecture, we are going to examine how pentose phosphate is regulated. It's regulated depending on the cell's need for ATP, NADPH, and ribose 5-phosphate. We can identify 4 different situations. In the first one, both ribose 5-phosphate and NADPH are needed by the cell. Next, more NADPH than ribose 5-phosphate is needed by the cell. Third possible situation is that more ribose 5-phosphate than NADPH is needed by the cell. And finally, both NADPH and ATP are needed by the cell, but ribose 5-phosphate is not. In the first case, when both ribose 5-phosphate and NADPH are needed by the cell, first 4 reactions of the pentose phosphate pathway predominate. That means that glucose 6-phosphate is oxidized to 6-phosphogluconodelta-lactone, which undergoes ring opening to 6-phosphogluconate, which is then oxidized to 3-oxo-6-phosphogluconate, decarboxylated to ribulose 5-phosphate. So those are first 3 oxidative reactions. And finally, fourth reaction is isomerization of ribulose 5-phosphate into ribose 5-phosphate. In the second case, if more NADPH than ribose 5-phosphate is needed by the cell, NADPH can be produced without synthesis of ribose 5-phosphate if ribose 5-phosphate is recycled to produce substrates for glycolysis. That means that ribose 5-phosphate undergoes series of reactions catalyzed by enzymes transketolases and transaldolases, which we just covered. And in those series of reactions, 6 molecules of ribose 5-phosphate produce 5 molecules of glucose 6-phosphate. In third case, if more ribose 5-phosphate than NADPH is needed by the cell, then ribose 5-phosphate is synthesized without the production of NADPH. That happens if oxidation steps of pentose phosphate pathway are simply bypassed. So then 2 molecules of fructose 6-phosphate and 1 molecule of glyceraldehyde are extracted from glycolysis and are utilized to make 3 molecules of ribose 5-phosphate. Enzymes again are transketolases and transaldolases that are described before. In the fourth, final case, both NADPH and ATP are needed by the cell, but ribose 5-phosphate is not. If that is the case, ribulose 5-phosphate produced in the pentose phosphate pathway is converted mainly to xylulose 5-phosphate and to a lesser extent to ribose 5-phosphate. Then those 2 pentoses in series of reactions catalyzed again by transaldolases and transketolases are converted to fructose 1,6-bisphosphate and glyceraldehyde 3-phosphate. Those are substrates for glycolysis, and they enter glycolysis to form pyruvate. Pyruvate then in turn is substrate for citric acid cycle, which we will cover a little bit later in this course. So pyruvate enters citric acid cycle where it produces additional ATP. So this completes our chapter on pentose phosphate pathway.